Okay, so ladies and gentlemen, you are welcome. So in today's video, we are going to look at the concept of ischemic acid pathway. And don't forget that if this is the first time you are coming to my YouTube channel, please join and subscribe the YouTube community. So introduction. The ischemic acid pathway is a crucial biosynthetic route in plants and microorganisms. So it is actually one of the biosynthetic pathways that take place in plants and microorganisms like protozoans, fungi, and algae. So this is actually the pathways that require or that usually take place in plant and microorganisms. And examples of these microorganisms include some bacteria, some algae, some protozoans, and fungi. And one very important thing in ischemic acid pathways, it involves the production of essential aromatic amino acids. So phenylalanine, tyrosine, and tryptophan are the major amino acids that are generated uh, through the ischemic acid pathway. So this pathway is significant for the synthesis of secondary metabolites. So it also used in the synthesis of secondary metabolites such as alkaloids and flavonoids. So you see, ladies and gentlemen, in ischemic acid pathway, it takes place in plant and microorganisms. It also involves the synthesis of aromatic amino acids with some secondary metabolites such as alkaloid and flavonoid. So what are the importance of the aromatic amino acids? So aromatic amino acids they play essential role in protein synthesis and hormone production. So of course, these three aromatic amino acids are among the 20 standard amino acids that are involved in protein synthesis. And of course, some of the proteins are hormones. So without the aromatic amino acids, it means that the hormones cannot be generated. So it means that the aromatic amino acid is important in protein synthesis and hormone production. They also serve as a precursor for numerous bioactive compounds that impact plant growth and development. So you see, apart from the synthesis of proteins and hormones, it also involves in synthesis of bioactive compounds. And some of these bioactive compounds they are very important in plant growth and development. And of course, the deficiency of this aromatic amino acid can lead to impaired physiological functions in both plant and animals. So it means that if there is any problem, there is any challenge with the synthesis of this aromatic amino acid, it affects the physiological functions in plants and also microorganisms. So you see, first is synthesize aromatic amino acid, and then second, it also involved in the synthesis of bioactive compounds that are essential for plant growth and development. And then also, it is deficiency can affect the physiology of all the organisms that undergo the ischemic acid pathway. And then the next thing is actually the overview. So ischemic acid pathway is actually a pathway that consists of 7 enzymatic steps. And it begins with the condensation of phosphoenol, pyruvate, and erythrospore phosphate. So you see here in the first step of the reaction, we have phosphoenol pyruvate, which condenes with d-erythrospore phosphate by an enzyme called 3-deoxy-D-arabinoheptulosonic acid 7 phosphate synthase. That is the enzymes that actually require to condense the two compounds, which is phosphoenol pyruvate and d-erythrospore phosphate. So this is the enzyme that contains the molecules together to generate 3-deoxy-D-arabinoheptulosonic acid 7 phosphate. So this is actually the product generated as a result of the condensation. So it is called DAO. Then the next one is actually another enzyme which is called 3-dehydroquinate synthase. So which actually lead to the production of dihydroquinic acids. So after the generation of 3-dihydroquinic acid, the next thing is actually the synthesis or the combustion of dihydroquinic acid to 3-hydroshikimic acid, that is DHS, using an enzyme called shikimate dehydrogenase. It is an enzyme that actually require NADP. Okay, so it is the NADP dependent enzymes. So after this generation of 3-dehydroshikimic acid, then the next one is actually generation of shikimic acid. So shikimic acid are produced, which subsequently in the actually presence of shikimate kinase generate shikimate 3 phosphate. So in the presence of an enzyme called 5-phenol pyrovyl shikimate 3 phosphate synthase is an enzyme that converts the shikimate 3 phosphate to the 5-enol pyrovyl shikimate 3 phosphates, and of course, which lead to the generation of chorismic acids in the presence of chorismate synthase. And of course, at the end, the chrosmic acid can be used in the synthesis of FABA, which is para-aminobenzoids. And of course, this further will lead to the generation of folic acid, that is folate, which is used as one of the sources of one carbon from carbohydrate, folate, which is a one carbon compound that can serve as a carrier of one carbon in this biosynthesis of molecules. And then after that, the chorismic acid can also lead to the generation of the phenates, which subsequently lead to generation of phenylalanine and tyrosine. And then also another product that can be generated from chorismic acid, we have anthranilites, which subsequently lead to the generation of tryptophan. So I can decide to ask the three metabolites that are generated from chorismic acid. So we have GABA, we have free phenates, and then we have anthranilites, which subsequently lead to the generation of aromatic amino acids. So shikimic acid is produced as an intermediate. So look at it, it is an intermediate. It is not the initial product and the final product, which subsequently lead to the synthesis of aromatic amino acid. So this shikimate will be ultimately the generation of the aromatic amino acid. So this is actually a brief description of shikimic acid pathway. It is a 7 enzymatic steps. The substrates of this pathway is phosphoenol pyruvate and erythro's 4-phosphate. And then the final product is phenylalanine, tyrosine, and tryptophan. And of course, the para-aminobenzoids, the phenates, and antranilites are the products that is generated from chorismic acid, which lead to the final production of the aromatic amino acids. So then the next thing is actually the key enzymes in the shikimic acid pathway. So each step of the pathway is catalyzed by specific enzymes that facilitate the transformation of substrates. So that is how we need, to see pathway. So in each of the pathway, you need to condense. So in each of the pathway, there's a specific enzymes which actually catalyze the transformational changes of the substrate to product. So the key enzymes in this pathway include 3-dehydroxy, D-arabinohexylosonate 7-phosphate synthase, and also the shikimate dehydrogenase. So these are the two important enzymes or key enzymes in this pathway. We have DAP synthase, and then we have shikimate dehydrogenase, which is an ADP dependent enzyme. So inhibitors of these two enzymes have been exploited for developing herbicides and antimicrobial agents. So, ladies and gentlemen, these are the two key enzymes. These are the two major regulatory enzymes in the shikimic acid pathway. So, if you can get any molecules that can inhibit the activities of these enzymes, it can serve as a herbicide because the pathway is usually taking place in plant and microorganisms. So, any compound that can inhibit the activity of these two enzymes, so it can serve as a herbicide. That is the molecules that can actually block the synthesis of the aromatic amino acid, which ultimately lead to the death of the plant. And then the next one is microbial agent. Because this pathway is how have been demonstrated to occur in microorganisms. So if you can get any molecules that can block the pathway, it can also block the synthesis of aromatic amino acids. So the next thing is the regulation of the pathway. So although we have different types of metabolic regulation, but we have like covalent modification, feedback inhibition, we have competitive inhibition, we have uncompetitive, mixed competitive. We have different types of inhibition as well as the regulatory mechanisms. So the regulatory mechanisms can be in the form of covalent modification. It can be in the form of allosteric regulation. And it can also be in a form of, we have so many of them. So, and we also have feedback inhibition. So the ischemic acid pathway is tightly regulated to ensure balance between the production and then the between the production of aromatic amino acid and their utilization. So feedback inhibition mechanisms is like actually types of mechanisms in which the end products can inhibit the activity of the upstream enzymes. Like for example, if you can now have the end product, let's say tryptophan inhibits DAHP enzymes, it means that you have a feedback inhibition, indicating that the end product of the pathway serves as the inhibitor of the starting enzymes of the pathway. That is actually described the feedback inhibition. So apart from the inhibitors, environmental factors such as light and nutrient availability, can also influence, can also affect the regulation of the ischemic acid pathway. So this is actually a ischemic acid. This is the formula of ischemic acid. So ischemic acid in pathway, what is the importance of ischemic acid in pathway? So ischemic acid is a precursor for the synthesis of antiviral drugs. So, like for example, oseltamivir, that is Tamiflu, is the ischemic acid pathway. So it can serve as an antiviral drug. It means that it can be used to kill virus. So it is actually extracted, this ischemic acid is extracted from the seeds of the Chinese star, which is an ice tree. This is where we usually extract the ischemic acid, which is a key sources of this compound. So research is ongoing on to explore the additional medicinal properties of ischemic acid and its derivatives. So you see here, we see that ischemic acid is having antiviral property. So then the next one is industrial applications. So the ischemic acids pathway has played a significant industrial applications, particularly in pharmaceuticals and agriculture. So you see in the pharmaceuticals how you can use the inhibitors of ischemic acid pathway to kill microorganisms or to actually kill herbicides. So it is used to produce bioactive compounds also that can serve as a natural pesticides or growth regulators. So in addition, apart from pharmaceutical and agricultural application, then we can get pesticide and growth regulators from the ischemic acid if you are able to identify and demonstrate how it competes into the enzyme's active site or into the pathway so that to actually kills the halves. So then the next one is actually the biotechnological advancements are aimed at enhancing pathway efficiency in microbial system for large-scale production. So in addition, actually, this pathway, if you can able to identify, so if you can see this, we can be able to identify that this pathway can be used to enhance the efficiency of the pathway in order to actually maintain the microbial life or to maintain the life of microorganisms for large scale production. Because this aromatic amino acid, they have so many applications. They have so many industrial applications like in food industry and pharmaceutical industry. So therefore, enhancing the pathway is very important in actually increasing the production of the aromatic amino acids. Then genetics engineering and synthetic biology. Advances in genetics engineering allow for the manipulation of the ischemic acid pathway in microorganisms. So therefore, synthetic biology approach enable the design of optimized pathways for increased yield of aromatic amino acids. So this technology holds promise for sustainable production method in various industries. So you see, in genetics engineering, it can be used to manipulate the ischemic acid pathway in microorganisms through gene editing or gene therapy. So you can edit the gene, or you can actually edit the enzymes in the pathway. You can up-regulate it or down-regulate it so that you can actually have the required amount of the ischemic acid in the microorganisms. While the synthetic biology is an approach that enables the design of optimized pathways for increased yield of aromatic amino acids. So see, both the genetics engineering and synthetic biology, they are just an approach that trying to increase the chances of the ischemic acid production. So these technologies hold promise for sustainable production method in various industries. So what are the challenges and the future direction? So despite it is important, the ischemic acid pathway faces challenges include metabolic bottlenecks. So future research is focused in improving pathway flux and understanding regulatory mechanisms. So there is a potential for discovering novel application of ischemic acid and its derivatives in various fields. So in conclusion, ischemic acid is essential for the biosynthetic of aromatic amino acid in plant and microorganisms, and it is significant spans agriculture, pharmaceuticals, and biotechnology highlighted its versatility. So ongoing research will continue to unlock the potential of this pathway for various industrial applications. So thank you very much, ladies and gentlemen. And if this is the first time, please try to join and subscribe to the YouTube community.